Beneath our feet, beyond our sight, lies a hidden kingdom that has shaped our world for over 1 billion years. It is a realm of silent architects and relentless recyclers, a network that connects all life in a vast subterranean web. This is the story of fungi. They are not plant, not animal, but a biological empire unto themselves. They were the first organisms to conquer the land, creating the very soil that would allow plants to grow. They have fed civilizations and brought them to their knees. They hold the power to heal our bodies with miraculous medicines and alter our minds with profound chemistry. From the yeast that leavens our bread to the intricate networks that sustain our forests, their influence is absolute, yet largely invisible. We are only now beginning to understand the true power of this ancient and mysterious kingdom, a force that governs the eternal cycles of growth, decay, and rebirth on planet Earth. Before the first leaf, before the first root, before any animals stirred on the continents, there was a silent conquest. Over 1 billion years ago, Earth was an alien world. The land was a vast expanse of sterile rock, baked by unfiltered solar radiation and scoured by relentless winds. The oceans teemed with simple life, but the continents were empty, hostile, and barren. To venture from the relative safety of the water onto this forbidding shore was a challenge of immense proportions. It required a form of life that was resilient, resourceful, and radically different from anything that had come before. That life was fungi. While primordial algae clung to the water's edge, the ancestors of modern fungi made the leap. They were not plants, nor were they animals. They were a kingdom unto themselves, equipped with a unique set of tools for survival. Their bodies were not solid structures, but vast, exploratory networks of microscopic threads called hyphae. Woven together, these threads form a mycelium, a living web that could probe and penetrate the smallest cracks in the rock. Unlike plants, they did not need sunlight. Instead, they were masters of chemistry, releasing powerful enzymes into their environment. These biological agents were capable of breaking down the very stone beneath them, prying loose precious minerals that no other organism could access. Their cell walls were not made of soft cellulose like plants, but of chitin, the same tough material that forms the exoskeletons of insects. This provided a durable defense against the harsh ultraviolet rays and extreme dryness of the terrestrial world. For hundreds of millions of years, these fungal networks were the dominant form of complex life on land. They were the planet's first miners and soil creators. As they dissolved rock and metabolized minerals, they combined this inorganic dust with their own organic matter when they perished. Slowly, painstakingly, thread by thread, they began to build the first thin layer of protosoil upon the naked stone. This was a planetary transformation on a geologic scale, executed by a kingdom of life that was almost entirely invisible. They were laying the foundation for a world they could not possibly have envisioned, preparing the ground for a revolution that would soon paint the barren continents green. The first conquerors had arrived, not with force but with patience and chemistry, forever changing the face of the earth. The stage was set. The fungal pioneers had created a foothold on the barren continents, a thin veneer of mineral-rich dust. But they held only one half of the key to terrestrial life. They could mine the earth, but they could not harvest the sky. They lacked the ability to perform photosynthesis, to turn sunlight into energy. Meanwhile, back in the sunlit coastal waters, green algae possessed this very power, but they were trapped. Without roots, they had no way to anchor themselves on land or to access the minerals and water locked away in the ground. Two kingdoms, each with a profound limitation, stood on the precipice of a world-changing partnership. The solution that emerged around 470 million years ago was one of the most significant events in the history of life. It was an alliance, a symbiosis so intimate that it continues to define nearly every ecosystem on Earth today. Fungi began to interface with the simple, rootless plants venturing onto the land. The fungal mycelium, already a master of subterranean exploration, extended its vast network far into the ground. These fine threads were vastly more efficient at absorbing water and mining for nutrients like phosphorus and nitrogen than any simple plant structure could be. The fungus would pass these essential elements directly into the plant's cells. In return, the plant, now anchored and nourished, would perform photosynthesis and share its energy-rich sugars with its fungal partner. This was the birth of the mycorrhizal relationship, a bond that enabled the greening of the globe. With fungi as their subterranean support system, plants no longer needed to remain tethered to the water. They could now push inland, climbing mountains and spreading across plains. This fungal-algal collaboration fueled an explosion of evolutionary innovation. Plants developed stems to reach for the sun, leaves to maximize its capture, and vascular systems to transport water. The planet's atmosphere began to change dramatically as these new green factories pumped out oxygen. The Earth was being wrapped in a green mantle, woven from sunlight by plants and stitched into the soil by the invisible threads of their fungal allies. It was a silent, cooperative takeover, a testament to the power of partnership, proving that the greatest leaps forward in evolution are not always born of competition, but of alliance. The partnership between plants and fungi was a staggering success. It fueled an evolutionary arms race, pushing plants to grow ever taller in their competition for sunlight. To achieve these new heights, plants developed a miraculous new material, lignin. This complex polymer, when integrated into cell walls, provided incredible strength and rigidity. It was the invention that allowed for the creation of wood, and with it, the first great forests on Earth. During the Carboniferous period, beginning around 360 million years ago, the world was covered in these forests. Vast, humid swamps were dominated by giant lycopod trees, reaching 100 feet into the sky, colossal ferns, and towering horsetails. The air grew thick with oxygen, reaching concentrations far higher than today, which in turn allowed insects to grow to startling sizes. But this new world had a profound and unforeseen problem. Lignin was a biological fortress. Its chemical structure was so complex and robust that nothing on Earth could break it down. The fungal kingdom, for millions of years the planet's master decomposers, had met its match. When these giant lignin-rich trees reached the end of their lives and fell into the swampy forest floor, they did not rot. They simply lay there, piling up one on top of the other, century after century, millennium after millennium. The great cycle of decay and renewal had been broken. For 60 million years, this process continued unabated. An immense amount of carbon pulled from the atmosphere by the plants was locked away in this undecayed wood. Instead of being released back into the environment, it was buried under layers of sediment compressed by time and geology. The planet was effectively burying its carbon. This massive sequestration created the vast coal seams that would one day fuel human industry. The Carboniferous world was a one-way street for carbon, from the air into wood and from wood into the ground. The fungi which had enabled the rise of these forests were now locked out, unable to perform their vital role. The entire planetary system was out of balance, waiting for the next great evolutionary leap from the hidden kingdom beneath the forest floor. For 60 million years, the Earth's carbon cycle was broken. The invention of lignin by plants had created a world where forests grew and fell, but never decayed, locking away their carbon in the ground. This geological stalemate could not last forever. The evolutionary pressure was immense. An entire planet's worth of energy, locked away in the unrotting wood of the carboniferous forests, was an opportunity waiting to be seized. The solution, when it came, was a biochemical revolution engineered by the fungal kingdom. Near the end of the Permian period, a new lineage of fungi evolved a weapon that would change the world forever. These fungi, the ancestors of the mushrooms we see today, developed a suite of powerful enzymes, most notably lignin peroxidase. This was the chemical key that could finally unlock the lignin fortress. The enzyme works in a remarkable way, initiating a cascade of reactions that dismantles the complex lignin polymer, breaking it down into simpler compounds that the fungi can absorb as food. For the first time, wood could be properly digested. The great logjam of the Carboniferous was over. Dead trees no longer piled up indefinitely. Instead, they became a source of nourishment, returning their stored carbon and nutrients to the ecosystem. This evolutionary innovation had profound consequences for the entire planet. The re-establishment of the decomposition cycle created a feedback loop. As nutrients were released from decaying wood, they fertilized the soil, promoting new growth. This kick-started the modern carbon cycle, where carbon flows from the atmosphere to plants to decomposers and back into the atmosphere and soil. It stabilized atmospheric oxygen levels, preventing them from rising to the dangerous highs of the Carboniferous. The era of coal formation on a massive scale came to an end. Fungi had reclaimed their title as the planet's essential recyclers. They were no longer just facilitators of growth, but also the arbiters of decay, the silent engineers who ensure that life's essential building blocks are never locked away forever. From that point forward, the forest floor would be a place not of permanent preservation, but of constant, dynamic transformation. The role of fungi as the great decomposers and plant partners only scratches the surface of their influence. Beneath our feet, in nearly every natural landscape on Earth, lies a hidden world of immense complexity and activity. It is the mycelial network, a vast, interconnected web of fungal threads that links the inhabitants of the forest into a single, cohesive community. This subterranean system has been called nature's internet, and for good reason. It is not merely a passive pipeline for nutrients, it is an active information highway that shapes the life of the entire ecosystem. Through these shared mycorrhizal networks, individual trees, even those of different species, are linked together. This connection allows for a level of cooperation that defies the traditional view of a competitive, tooth-and-claw natural world. A large, sun-drenched parent tree, for instance, can send excess sugars through the fungal network to nourish smaller saplings struggling in the shade. It's a form of community support, ensuring the health and resilience of the forest as a whole. But the network transmits more than just resources. It also carries information. When one tree is attacked by harmful insects, it can release chemical distress signals into the mycelial web. These signals travel to neighboring trees, which then ramp up their own chemical defenses in preparation for the impending attack, a forest-wide immune response coordinated by their fungal intermediary. This intricate web of relationships extends to the animal kingdom as well. Certain species of ants have become farmers, cultivating specific fungi in subterranean gardens for food. Mammals, like wild boars and squirrels, have a deep connection with fungi that produce underground fruiting bodies such as truffles. By digging up and consuming these delicacies, the animals help to disperse the fungal spores over a wide area. The forest is not a collection of individual organisms, but a single integrated superorganism, and the mycelial network is its nervous system. It is a world of constant communication and exchange, a dynamic marketplace of resources and information. Understanding this hidden kingdom is to understand the true nature of life on Earth, not as a story of individuals, but as a story of connection, collaboration, and ancient, unbreakable alliances. Imagine a world of naked rock, a planet forged in fire scoured by wind and water, but utterly devoid of soil. This was the early Earth, a sterile landscape waiting for a transformation. The agent of that change was not a mighty plant or a complex animal, but a humble and profoundly strange partnership. This is the story of lichen, the ultimate pioneer. Lichen is not a single organism, but a composite, a stable symbiotic association between a fungus and an alga, or cyanobacterium. The fungus provides the structure, a protective house that shields its partner from harsh sunlight and dehydration. In return, the photosynthetic partner provides food, converting sunlight into energy. Together, they form an entity far more resilient than either could be alone. They are the first to arrive and the last to leave, clinging to life in the most inhospitable places on Earth, from scorching desert rocks to the frozen plains of Antarctica. Their true power, however, lies not just in their survival, but in their ability to create. As lichen establishes a foothold on bare rock, the fungal partner begins its slow, patient work. It secretes a cocktail of powerful acids, chemical marvels that etch the stone itself. Over centuries and millennia, this gentle but relentless assault breaks the rock down, grain by grain, creating the first rudimentary dust of what will one day become soil. This process, known as biological weathering, is the foundation of nearly all terrestrial ecosystems. Once the lichen has done its work, creating a thin layer of organic matter and mineral dust, other, slightly less hardy organisms can move in. Mosses take root in the shallow substrate. Insects and tiny invertebrates find shelter and sustenance. The death and decay of these newcomers add more organic material, deepening the nascent soil. It is a chain reaction of creation, started by the quiet, persistent labor of a fungus and its microscopic partner. They are living testaments to the power of cooperation, capable of surviving the vacuum of space and the harshest climates on our own world. They are the painters of the wilderness, splashing vibrant colors across stark granite canvases. But most importantly, they are the world builders, the silent architects who turned a planet of sterile rock into a world of green, vibrant life. This profound ability of fungi to transform their environment, to create and build, also concealed a darker potential, one that would directly impact the course of human history. In the cool, damp climates of medieval Europe, rye was the grain of the people. It was hardy, reliable, and formed the basis of their daily bread. But within its dark, heavy grains, a sinister stowaway sometimes took hold. It appeared as a dark, purplish, horn-like growth, easily mistaken for a discoloured kernel. This was ergot, the sclerotium of the fungus Claviceps purpurea. When unknowingly harvested and milled into flour, it unleashed a terrifying affliction upon entire communities. The condition, now known as ergotism, manifested in two horrific forms. The first was convulsive, a torment of the nervous system. Victims would suffer from uncontrollable spasms, contortions, and vivid, terrifying hallucinations. They spoke of crawling sensations under their skin and visions of otherworldly beings. Entire villages seemed to go mad, leading to outbreaks of what was called the dancing plague. Without a scientific explanation, the populace turned to superstition, blaming demonic possession or witchcraft. Historians now speculate that the bizarre testimonies and behaviours during the 1692 Salem witch trials may have been fuelled in part by an outbreak of ergotism from contaminated rye. The second form of the affliction was gangrenous, known with dread as St. Anthony's fire. The alkaloids in the fungus are powerful vasoconstrictors, meaning they drastically tighten blood vessels. This cuts off circulation to the extremities, causing an agonizing burning sensation in the limbs. The afflicted felt as if they were being consumed by an inner fire. The lack of blood flow would eventually cause the tissues in the hands and feet to decay, leading to a loss of limbs. The name came from the monks of the Order of St. Anthony, who became specialized in caring for the sufferers. For centuries, these outbreaks were a mysterious scourge, a divine punishment or a devil's curse baked into the very bread meant to give life. It was a fungal blight that drove people to madness, shaped folklore, and may have even influenced the outcomes of famous historical trials. The fungus demonstrated a terrifying power to manipulate the human mind and body. Yet, within its toxic chemistry lay a profound secret. The very compounds that caused such misery would one day be isolated and studied, yielding potent pharmaceuticals for everything from migraines to childbirth, forever linking this medieval terror to the birth of modern pharmacology. Long before humans understood the science of microbiology, they had formed a powerful alliance with the fungal kingdom. This partnership was not born in a laboratory but in kitchens, bakeries, and breweries through a process that seemed like everyday magic: fermentation. The star of this story is yeast, a humble, single-celled fungus with an insatiable appetite for sugar. For millennia, its work was invisible, its existence unknown, yet its impact on civilization is immeasurable. Consider bread, the staff of life. For early agricultural societies, a paste of ground grain and water resulted in a dense, flat, and often tough cake. But when wild yeasts from the air settled on the dough, a miracle occurred. The yeast consumed the sugars in the flour, releasing tiny bubbles of carbon dioxide gas. These bubbles became trapped in the dough's structure, causing it to rise, becoming light, airy, and more digestible. Leavened bread was not just more palatable, it was a more efficient way to deliver calories, a foundational technology that fueled the growth of cities and empires. The same microscopic organism was also responsible for another of humanity's oldest traditions, brewing. When yeast was introduced to a mash of sugary grains or fruit juice, it produced not just carbon dioxide, but alcohol. In ancient Mesopotamia, Sumerians sang hymns to Ninkasi, the goddess of beer. For them, beer was a divine gift. It was a social lubricant, a ritualistic staple, and often a safer alternative to contaminated water sources. Wine, too, owes its existence to the yeasts naturally present on grape skins. This fungal transformation didn't stop there. In the world of cheese, molds from the Penicillium genus work their way through curds, creating the sharp, complex flavors and iconic blue veins of Roquefort and Gorgonzola. In Asia, the mold Aspergillus oryzae is the indispensable workhorse used to ferment soybeans, creating the savory, umami-rich flavors of soy sauce and miso. Fungi gave us more than just delicious food and drink. They gave us preservation. Fermentation allowed humanity to transform perishable harvests into stable products that could be stored through winter, preventing famine and enabling long-distance travel. These gifts nourished our bodies, shaped our cultures, and brought people together in celebration and daily ritual. But this intimate knowledge of the fungal kingdom was a double-edged sword. For every beneficial mold that gave us bread, another was waiting with a far more sinister capability. In the gilded halls of power, where ambition and paranoia walked hand in hand, a silent and sophisticated weapon has always been prized, one that is subtle, untraceable, and deadly. For centuries, the fungal kingdom offered just such a tool, hidden in plain sight. This is the story of the mushroom as the poisoner's perfect accomplice. Among the thousands of mushroom species, a few contain some of the most potent toxins known in nature. The most infamous are the Amanita family, which includes the deceptively beautiful deathcap and the starkly white destroying angel. Their danger lies in their treachery. They often resemble edible varieties and by all accounts have a pleasant, mild taste. There is no bitter warning, no immediate sign of the fatal mistake that has been made. The true horror of their toxins, known as amatoxins, is the delay. After being ingested, the amatoxins begin their methodical work, targeting the core machinery of life itself by halting protein synthesis in the liver and kidneys. Yet, for 6, 12, or even 24 hours, the victim feels perfectly fine. This asymptomatic period is what made the mushroom such a formidable weapon for assassins. By the time the first violent symptoms appear, the toxins have been fully absorbed, and the damage is irreversible. The most famous alleged victim of this fungal intrigue was the Roman Emperor Claudius in 54 AD. Historical accounts, most notably from the historian Tacitus, suggest that his ambitious wife Agrippina the Younger served him a dish of tainted mushrooms. Her goal was to clear the path to the throne for her own son, Nero. Claudius's passing, which occurred after a period of intense suffering, was conveniently timed. In an age without forensic science, the cause was attributed to natural illness, and Agrippina's scheme succeeded. This was not an isolated case. Centuries later, in 1740, Holy Roman Emperor Charles VI died a week after consuming a meal of deathcap mushrooms. His demise left no direct male heir, plunging the continent into the devastating War of the Austrian Succession. A single fungal meal redrew the map of Europe. The mushroom's ability to deliver a quiet, delayed, and agonizing end cemented its reputation in folklore as a symbol of dark magic and betrayal. It was the poisoner's kiss, a gift from the earth that could topple empires. But from another, unassuming corner of this same kingdom, a different kind of silent power was about to emerge, not to end life, but to save it on a scale previously unimaginable. The battlefields of the First World War were a testament to the limits of medicine. Soldiers survived grievous injuries only to succumb days later to rampant infection. Doctors could set bones and stitch wounds, but against the invisible onslaught of bacteria, they were virtually powerless. Sepsis and gangrene were the true victors of the conflict. A decade later, in a cluttered London laboratory, a moment of serendipity would provide humanity with its greatest weapon in this ancient war. In September 1928, bacteriologist Alexander Fleming returned to St. Mary's Hospital after a holiday. He began sorting through a stack of petri dishes containing cultures of Staphylococcus bacteria. One dish caught his eye. It was contaminated with a spot of bluish-green mold, a common lab nuisance. But Fleming, a notoriously untidy but brilliant observer, noticed something extraordinary. In a clear circle surrounding the mold, the bacteria had been completely wiped out. He realized the mold was producing a substance that was lethal to the bacteria. He cultured the mold, identified it as being from the Mycelium genus, and named the bacteria-inhibiting substance he extracted penicillin. Fleming had made a monumental discovery, but he could not overcome the next great hurdle, producing a stable, purified, and concentrated form of the drug. For over a decade, penicillin remained a scientific curiosity, a footnote in medical journals. The world was on the brink of another global conflict when a team at Oxford University, led by Howard Florey and Ernst Chain, resurrected Fleming's work. With Britain under siege, they worked relentlessly. In their quest to grow enough mould, they improvised with any vessel they could find, including hospital bedpans and milk churns. In 1941, they were ready for their first human test. The patient was Albert Alexander, a police constable with a severe infection. The initial injections of penicillin had a miraculous effect. His fever dropped, and he began to recover. But their meagre supply ran out after 5 days, the infection returned, and he ultimately passed away. The tragic outcome was also a stunning proof of concept. The challenge was now production. The project moved to the United States, where government and private industry launched a massive effort. A breakthrough came when a more potent strain of Penicillium was found on a mouldy cantaloupe in a market in Peoria, Illinois. By D-Day in 1944, Allied forces had enough penicillin to treat all their wounded, saving thousands of lives. A chance observation of a common mold had ushered in the age of antibiotics, fundamentally transforming medicine and giving humanity a fighting chance against its oldest microscopic foes. In the early 1840s, Ireland was a nation on a knife's edge. Its population had swelled to over 8 million people, a significant portion of whom were impoverished tenant farmers. For them, survival depended almost entirely on a single crop, the potato. It was a nutritional miracle, capable of feeding a family for a year on just a small patch of land. This reliance, however, created a fragile monoculture, a biological house of cards waiting for the right gust of wind to topple it. That wind arrived in the summer of 1845. It came not as a storm, but as a strange, silent fog that rolled in from the coast. Farmers reported a peculiar odour in the air, a smell of decay, where there should have been the earthy scent of harvest. Within days, the leaves of their potato plants began to wither and turn black. When they dug into the soil, desperate for their food source, they found only a foul-smelling, inedible mush. The culprit was an organism they could not see and did not understand, Phytophthora infestans, a water mould, a fungus-like protist. Its spores had travelled across the Atlantic, likely in the holds of ships from the Americas. In the cool, damp conditions of the Irish summer, it found a perfect incubator. The blight spread with terrifying speed, wiping out nearly half the potato crop in its first year. The next year, it was a total failure. The consequences were catastrophic. What followed was not just a famine, but a complete societal collapse known as the Great Hunger. With their sole food source gone, millions faced starvation. Landlords, many of them absentee English aristocrats, continued to demand rent, evicting families who could no longer pay. Food was still being produced in Ireland, grain, cattle and dairy, but it was exported for profit, while the Irish people starved. The response from the British government was tragically inadequate, hampered by bureaucracy and a prevailing belief that interfering with the market was wrong. Soup kitchens were too few, and public works projects often demanded more physical strength than the starving population could muster. Over the next 5 years, the nation was hollowed out. An estimated 1 million people perished from hunger and related diseases. Another million were forced to emigrate, boarding crowded, disease-ridden vessels, later called coffin ships, in a desperate flight for survival. This microscopic organism did more than destroy a crop. It redrew the demographic, political and cultural map of Ireland forever. It left a legacy of loss and resentment that would echo for generations, a horrifying testament to how the fate of a nation can be sealed by the arrival of a single, unseen spore. Long before the modern world began its scientific inquiry into the mind, ancient cultures were already exploring the far reaches of consciousness. Their guides on these journeys were not philosophers or physicians, but humble fungi sprouting from the forest floor. In Mesoamerica, the Aztecs called them Teonanacatl, or flesh of the gods. Carved stone effigies dating back 3,000 years depict figures crowned with mushrooms, suggesting a deep, sacramental relationship between humanity and psychoactive fungi. For centuries, this knowledge remained hidden from the Western world, dismissed as folklore or relegated to the notes of Spanish friars who condemned the practices. The secret was kept by indigenous communities like the Mazatec people of southern Mexico, who continued their sacred mushroom ceremonies in seclusion. That changed in 1955. An American banker and amateur mycologist named R. Gordon Wasson traveled to Oaxaca, driven by a lifelong fascination with the cultural role of fungi. There, he was introduced to the shaman Maria Sabina, who, after much persuasion, allowed him to participate in a nighttime ritual. In her candlelit hut, Wasson consumed the psilocybin mushrooms and experienced what he described as a profound, world-altering journey. His 1957 article in Life magazine titled "Seeking the Magic Mushroom" introduced this secret world to millions. The floodgates were opened. The article captured the imagination of a generation on the cusp of cultural revolution. Figures like psychologist Timothy Leary at Harvard began researching psilocybin for its psychological effects, though his methods soon strayed from rigorous science into cultural advocacy. The mushrooms became a symbol of the 1960s counterculture, fueling a new wave of art, music, and social exploration. For decades, research was stifled by legal restrictions and social stigma, but today, we are in the midst of a scientific renaissance. Neuroscientists at leading universities are using advanced imaging technology to study how these compounds affect the brain. They've found that psilocybin can decrease activity in the brain's default mode network, which is linked to our sense of self, potentially leading to feelings of interconnectedness and ego dissolution. This research is showing remarkable promise for treating conditions like depression, anxiety, and addiction. We are only just beginning to scientifically understand what ancient cultures seem to know intuitively, that certain fungi hold a key, a chemical code that can unlock new perspectives on the mind, our world, and our place within it. In the rainforests of the world, a silent, microscopic drama unfolds every day. An ant, foraging on the forest floor, moves with purpose. But then its behavior changes. It abandons its colony and its duties, compelled by an unseen force to climb. It ascends a stalk of vegetation, moving against all its natural instincts until it reaches a specific height and orientation. There, it clamps its mandibles onto the underside of a leaf in a final, convulsive act. It will never let go. The ant is no longer in control. It has become a puppet, and the puppeteer is a fungus. This is the world of cordyceps, a genus of parasitic fungi with hundreds of species, each highly specialized to infect a particular type of insect. The process begins when a microscopic spore lands on an unsuspecting host. It germinates, releasing enzymes that breach the insect's tough exoskeleton. Once inside, the fungus begins to grow, its thread-like mycelium weaving through the host's body, consuming its non-essential tissues from the inside out. For a time, the fungus keeps its host alive, a living incubator for its own proliferation. The most remarkable stage is the manipulation. The fungus produces a cocktail of unique chemical compounds that interact with the host's nervous system. It hijacks the insect's brain and muscles, compelling it to perform its final, bizarre pilgrimage. This zombie behavior is not random, it is a calculated strategy. The fungus maneuvers its host to a location with the precise temperature and humidity it needs to reproduce. Once the ant is locked in its final position, the fungus delivers the final blow, and the host's life ends. Soon after, the next stage of the fungus's life begins. A fruiting body, a vibrant stalk called a stroma, erupts from the back of the dead insect's head, growing upwards towards the light. From the tip of this stalk, it will release thousands of new spores, which rain down onto the forest floor below, ready to infect the next wave of unsuspecting victims. While its life cycle is the stuff of science fiction, cordyceps has also found a place in human culture, particularly in traditional Chinese and Tibetan medicine, where it is highly valued for its purported health benefits. It is a stark reminder that in the fungal kingdom, survival takes many forms, from partnership to predation, and even to the complete and total mastery of another creature's will. When we walk through an ancient forest, we see a collection of individuals, a towering oak, a slender birch, a young sapling, all seemingly competing for sunlight, water, and space. We see the struggle for survival playing out above ground. But we are missing the bigger picture. Beneath our feet, hidden in the soil, lies a bustling, vibrant community, a complex network that connects nearly every plant in the forest. This is the mycorrhizal network, an information superhighway made of fungi. The relationship is one of the oldest and most successful partnerships on Earth, a symbiosis that allowed plants to first colonize land over 400 million years ago. The bargain is simple. Plants, through photosynthesis, create sugars and carbon. Fungi, however, cannot produce their own food, so their microscopic threads, called hyphae, fuse with the tips of plant roots. The plant channels up to 30% of its sugars down to the fungus. In return, the fungus acts as a vast extension of the plant's root system. A single fungus can be enormous, its network of hyphae stretching for miles, far more efficient at mining the soil for essential nutrients like phosphorus and nitrogen and absorbing water, which it then provides to the plant. But this is more than just a simple trade. The fungal hyphae from one tree connect with the hyphae from another, linking them together in a shared grid. This wood wide web, a term coined by scientists, allows for incredible interactions. Ecologist Suzanne Simard's groundbreaking research revealed that trees use this network to communicate. If one tree is attacked by insects, it can send chemical distress signals through the fungal network to its neighbors, which then ramp up their own defenses. It is also a resource-sharing system. Large, well-established mother trees act as central hubs, using the network to shuttle excess carbon and nutrients to shaded seedlings, including their own offspring, giving them a better chance at survival. A forest is not a collection of solitary individuals, it is a cooperative. It functions like a single intelligent organism, with the fungal network acting as its distributed brain and circulatory system. This discovery fundamentally changes our perception of nature. It reveals a world of collaboration, communication, and mutual support happening silently and invisibly beneath the soil, a hidden architecture that sustains the entire forest ecosystem. Recognizing this interconnectedness is vital, for if we damage this delicate web, we risk the health of the very forests on which our planet depends. The soil beneath our feet is one of the most densely populated habitats on Earth. A single teaspoon can contain billions of microorganisms engaged in a constant, unseen struggle for resources. In this microscopic jungle, there are grazers, decomposers, and prey. And where there is prey, there are always predators. We often think of fungi as passive decomposers, patiently breaking down dead organic matter. But some species have evolved a more aggressive strategy. They are hunters, and their preferred quarry is one of the most abundant animals on the planet, the nematode, or roundworm. These predatory fungi have developed an astonishing array of sophisticated traps to capture their fast-moving prey. One of the most remarkable is the constricting ring. A fungus like Arthrobotrys oligospora grows dozens of these microscopic lassos from its hyphae. Each ring is composed of 3 cells. When an unsuspecting nematode wriggles through the opening, it triggers a response. In less than 1/10 of a second, the 3 cells inflate with water, swelling inwards and trapping the worm in an inescapable grip. It is one of the fastest cellular movements in biology. Other fungi employ different tactics. Some produce sticky, 3-dimensional nets or knobs, their surfaces coated with an adhesive that functions like biological superglue. A nematode that brushes against it is immediately stuck fast. The more it struggles, the more entangled it becomes. Still, others engage in a form of chemical warfare, secreting toxins that paralyze any nematodes in the vicinity, leaving them helpless for the fungus to approach and consume. Once the prey is captured, by whatever means, the next stage begins. The fungus pierces the nematode's outer layer and grows specialized hyphae into its body. It releases digestive enzymes, turning the worm's internal structures into a nutrient-rich soup, which it then absorbs. For the fungus, the nematode is a vital source of nitrogen, an essential element that can be scarce in decaying wood or soil. These microscopic hunters play a crucial ecological role, helping to regulate nematode populations that might otherwise damage plant roots and harm agricultural crops. Their existence reveals yet another facet of the fungal kingdom's incredible adaptability. They are not just the world's recyclers or its great connectors. In the dark, hidden world of the soil, they are also among its most patient, efficient, and ingenious predators. Deep within the world's oldest forests, long after the sun has set, an otherworldly glow emanates from the forest floor. This is not the work of fireflies nor a trick of the moonlight. This is foxfire, a spectral luminescence produced by the kingdom of fungi. For centuries, this phenomenon has fueled folklore and superstition, tales of wandering spirits and enchanted woods. Mark Twain even used its ethereal light to illuminate a dramatic scene in The Adventures of Huckleberry Finn. But behind the myth lies a profound biological mystery. Why would a fungus, an organism rooted in place, need to create its own light? The answer is a marvel of biochemistry. The glow is the result of a chemical reaction involving a compound called luciferin and an enzyme, luciferase. When they combine with oxygen, energy is released in the form of a cold, green light. While over 80 species of glowing fungi have been identified, their reasons for doing so remain a subject of intense scientific debate. One leading theory suggests the light is a beacon. At night, it attracts insects and other creatures. These visitors crawling over the mushroom inadvertently pick up spores and carry them to new locations, helping the fungus reproduce and spread. It is a silent, luminous advertisement in the complete absence of sunlight. Another hypothesis posits the opposite, that the glow serves as a warning, deterring nocturnal animals that might otherwise consume the fungus. It could be a signal that says, "I am not your food." A more recent and perhaps less romantic idea is that the light is simply an accidental byproduct of the fungus's metabolic processes, specifically those involved in breaking down lignin, the tough polymer, in wood. As the fungus digests its meal, the chemical reactions produce a faint but constant glow from the mycelial network threading through the decaying logs. From the ghostly sheen of the honey mushroom's vast underground networks to the delicate, glowing caps of the ghost fungus, this natural illumination represents a frontier in biotechnology. Scientists are studying its genetic basis, hoping to harness this natural light for everything from medical imaging to self-illuminating trees that could one day light our city streets. The forest's secret lanterns are more than just a beautiful spectacle. They are a living puzzle, a testament to the elegant and often enigmatic solutions that evolution can produce. If fungi can conjure light from darkness, what other incredible abilities are they hiding just beyond our sight, waiting for us to understand? Our modern world is scarred by its own progress. Industrial sites seep toxins into the soil, oil spills stain our waters, and persistent chemicals accumulate in the environment, creating wounds that seem impossible to heal. Yet, in these blighted landscapes, an ancient and powerful ally is being deployed. This is the science of mycoremediation, a revolutionary approach that uses the innate abilities of fungi to cleanse and restore our planet. The secret to this power lies in the mycelium, the vast, branching network that forms the true body of the fungus. To feed, mycelium secretes a cocktail of powerful enzymes into its surroundings. These are the same enzymes that allow fungi to break down the most resilient materials in nature, like the lignin and cellulose in wood. It turns out that many of our most stubborn pollutants, from petroleum hydrocarbons to pesticides and industrial solvents, have chemical structures remarkably similar to these natural compounds. The fungi do not distinguish between a fallen log and a diesel spill. They simply see a source of food. They unleash their enzymatic toolkit, breaking down complex, toxic molecules into simpler, harmless components. The pioneering work of mycologist Paul Stamets dramatically demonstrated this potential. In one experiment, he inoculated piles of diesel-contaminated soil with the mycelium of oyster mushrooms. Within weeks, the mushrooms had digested the oil, breaking it down into water, carbon dioxide, and carbohydrates. The soil was not only cleaned but revitalized, bursting with new life, insects, plants, and then birds. Fungi's healing touch extends beyond oil. Certain species are hyperaccumulators, capable of absorbing and concentrating heavy metals like mercury, lead, and cadmium from polluted soil and water, effectively filtering the environment. In the wake of the Chernobyl disaster, researchers discovered species of radiotrophic fungi growing inside the damaged reactor, seemingly drawn to the radiation as a source of energy. This remarkable adaptability opens up possibilities we are only just beginning to explore. While not a cure-all, mycoremediation offers a powerful, low-cost, and sustainable biological partnership. It represents a fundamental shift in our approach to pollution, moving from containment to active, biological restoration. The same kingdom that has managed the planet's decomposition cycles for 1 billion years is now offering us a chance to clean up our own mess, one mycelial thread at a time. For millennia, humanity has built its world by quarrying stone, felling forests, and forging metal. Our civilization is constructed from materials that are extracted, processed, and assembled at a tremendous environmental cost. But a revolution is quietly growing in labs and workshops around the world, one that proposes a radical new way to build, not by manufacturing, but by cultivating. The architect of this future is Mycelium. By harnessing the natural growth of this fungal network, we can create a new class of materials that are strong, versatile, and completely compostable. The process is elegant in its simplicity. Agricultural waste products like sawdust, corn husks, or hemp fibers are sterilized and then inoculated with a chosen fungal species. Packed into a mold of any desired shape, a brick, a panel, a piece of packaging, the mycelium begins to grow. Over several days, it weaves its microscopic filaments through the substrate, binding it together into a solid, lightweight, composite material. Once the object has reached the desired density, it is gently heated to stop the fungus's growth, resulting in a finished product that is inert and durable. These mycomaterials have astonishing properties. They are excellent thermal insulators, naturally fire-resistant, and can possess a compressive strength greater than concrete. Companies like Ecovative Design pioneered this technology, creating protective packaging that serves as a direct, biodegradable replacement for plastic foams like styrofoam. But the applications go far beyond packaging. Architects and designers are now exploring mycelium for everything from acoustic panels and furniture to entire building structures. Philip Ross, an artist and inventor, has grown bricks and chairs, demonstrating the material's structural potential. Projects like the Growing Pavilion showcase entire temporary structures built from mycelium panels, illustrating a future of organic, living architecture. Even NASA is investigating the use of fungi to grow habitats on Mars, envisioning a future where astronauts can cultivate their own shelters from a small, transportable block of dormant mycelium. This is more than just a new material. It is a new philosophy. It is a shift from an extractive economy to a regenerative one, where our buildings are grown from local waste streams and, at the end of their life, can be returned to the earth to enrich the soil. The fungal kingdom is offering us a blueprint for a circular, sustainable civilization, one that is literally grown from the ground up. We have mapped the distant galaxies, plumbed the ocean's deepest trenches, and sequenced our own genome. We consider ourselves masters of a known world. Yet, We remain almost completely ignorant of a biological empire that underpins nearly every ecosystem on the planet. For every species of plant, there are at least 6 species of fungi. Current scientific estimates suggest that our world is home to as many as 3.8 million fungal species, a number that dwarfs the diversity of plants and rivals that of insects. The shocking reality is that we have formally identified and named fewer than 10% of them. More than 90% of the fungal kingdom remains a complete mystery, a vast, unexplored continent of biological dark matter. These undiscovered organisms are not just hiding in remote, inaccessible jungles. They are everywhere, in the soil of our backyards, living inside the leaves of every tree, thriving in the ice of Antarctic glaciers, and colonizing the deepest hydrothermal vents. The challenge is that most of them are microscopic and cannot be grown in a laboratory setting. For centuries, mycologists were limited to studying what they could see and cultivate. But the advent of modern genetics has thrown the doors to this hidden world wide open. Using techniques like environmental DNA or eDNA, scientists can now take a sample of soil or water and sequence all the genetic material within it. This reveals the presence of hundreds, sometimes thousands, of fungal species that have never been seen before. Each new discovery is a potential breakthrough. Within this undiscovered majority lie the solutions to some of humanity's greatest challenges. They could be the source of the next generation of life-saving antibiotics, powerful new enzymes for industrial processes, or novel compounds to create sustainable biofuels. Some may be key symbiotic partners for agricultural crops, offering a way to grow food with fewer chemical fertilizers. But we are in a race against time. As climate change alters environments and deforestation destroys habitats, we are causing a silent extinction. We are losing countless species of fungi before we even have a chance to know they exist, erasing millions of years of evolutionary wisdom. The great scientific expeditions of the 21st century may not be to other planets, but into the soil beneath our feet. The quest to map this immense, unknown kingdom is one of the most urgent and important undertakings of our time. Our journey through the Earth's hidden kingdom has revealed a world far more complex and influential than we ever imagined. For over 1 billion years, fungi have been the silent architects of our planet, the great decomposers and connectors, driving evolution and shaping the world we inhabit today. They are not merely a curious branch on the tree of life, they are its root system. Now, as we face the unprecedented challenges of the 21st century, this ancient kingdom is emerging as a vital partner for our future. The solutions they offer are as diverse as the kingdom itself. We have seen how their enzymatic power can heal polluted lands, and how their mycelial bodies can be grown into sustainable materials to build our homes and replace our plastics. This is the foundation of a new, circular economy, one modelled on the fungal principles of decomposition, regeneration, and interconnection. But the frontiers of mycology extend even further into realms that were once the domain of science fiction. Researchers are now exploring the information processing capabilities of mycelial networks. These living webs behave like biological computers, sensing their environment, transmitting information, and making decisions in a decentralized way that is both resilient and efficient. This could inspire revolutionary new forms of computing and network design. In the field of medicine, the fungal compound psilocybin is at the forefront of a renaissance in mental health research, offering new hope for conditions that have long resisted treatment. It seems fungi have the potential to re-engineer not just our ecosystems, but our own consciousness. To fully embrace this fungal future requires a profound philosophical shift. It demands that we move past a human-centric worldview and recognize our deep, unbreakable interdependence with the living world around us. We are not separate from nature. We are one small part of a vast, interconnected web, and fungi are the master weavers. They teach us that resilience comes from connection, that decay is essential for renewal, and that collaboration is a more powerful force than competition. The story of fungi is ultimately the story of life's persistence. It is a story written in every handful of healthy soil, in every forest, and in every breath we take. As we learn to listen to their ancient wisdom, we may find that the keys to a sustainable and even thriving human future have been beneath our feet all along.